Imagine being able to rewrite the code of life, to cure genetic diseases or create new species. This is the power of CRISPR, a revolutionary gene editing tool that's changing our world. It's like a molecular scissors, cutting and pasting DNA sequences with unprecedented precision. The potential is mind-boggling, from curing genetic diseases to enhancing crop yields, even to the creation of new species. The implications are vast, and we are just scratching the surface. Welcome to the fascinating world of CRISPR. Before we delve deeper into the world of CRISPR, it's important to understand the journey of gene editing. It began with the discovery of the DNA double helix structure by Watson and Crick in 1953, a breakthrough that revolutionized our understanding of life's blueprint. This laid the foundation for the development of early gene editing tools, like zinc finger nucleuses and TALENs. However, these tools were often expensive, difficult to work with, and lacked precision. The world of gene editing was in need of a simpler, more efficient tool. Then came CRISPR, a game-changer that was poised to turn the world of genetics on its head. CRISPR isn't just a fancy acronym, it's a game-changer in the field of genetics. Decoding this acronym, we have clustered regularly interspaced short palindromic repeats. Quite a mouthful, isn't it? Discovered as a part of bacterial DNA, these sequences are a key player in their immune system, helping bacteria fend off viral invaders. Now imagine if we could harness this bacterial defense mechanism for our own genetic engineering. Intriguing, right? But how can bacteria's defense mechanism change our lives? Before diving deeper into how CRISPR works, let's take a step back and understand how this revolutionary tool was discovered. CRISPR was first identified in the late 1980s, buried within the genetic material of a simple bacteria. Scientists found these strange DNA sequences, clustered regularly interspaced short palindromic repeats, hence the name CRISPR. But it wasn't until the early 2000s that they understood its purpose. It was a part of the bacteria's immune system, a defense mechanism against viral invaders. Upon this discovery, scientists realized the potential of using this system for human genetic engineering, a tool that could change the face of medicine and biology. This was a breakthrough that opened up a whole new world of possibilities in the field of genetics. To comprehend CRISPR's power, we need to understand its main component, Cas9. At its core, Cas9 serves as a pair of molecular scissors, capable of cutting strands of DNA at specific locations. This ability to cut DNA lies at the heart of CRISPR's gene editing prowess. But how does Cas9 know where to cut? The answer lies in guide RNA, a molecule that partners with Cas9. This guide RNA is designed to match a specific DNA sequence in a gene that we want to edit. When the Cas9 complex encounters this sequence, it recognizes its target and makes the cut. Now imagine the human genome as a vast library and you're looking for a specific word in a specific book. With its guide RNA, Cas9 acts as a highly accurate GPS that leads you straight to your destination. And once it gets you there, it performs a precise cut. In essence, CRISPR-Cas9 is a precise genetic GPS and scissors combined. Let's take a closer look at how CRISPR works. The CRISPR system, with its Cas9 and guide RNA, is like a search and destroy mission on a microscopic scale. Once the Cas9 and guide RNA complex locates and cuts the target DNA sequence, the cell's natural repair mechanisms kick in. If we want to replace the cut sequence with a different one, we can introduce a new DNA sequence into the cell. The cell's repair machinery will incorporate this new sequence in place of the cut DNA. It's like cutting a sentence out of a book and seamlessly pasting in a new sentence. The precision and efficiency of CRISPR, combined with its ability to introduce new genetic material, make it a powerful tool in genetics. It's this mechanism that opens up a world of possibilities, from curing genetic diseases to enhancing crop yields. With such precision, the applications of CRISPR-Cas9 are virtually limitless. Imagine a world where genetic diseases, the bane of many lives, are a thing of the past. This isn't a far-off future. CRISPR technology could make it possible to edit out disease-causing mutations from our DNA, offering a cure for conditions like cystic fibrosis or sickle cell anemia. But the potential of CRISPR doesn't stop at human health. It's also making waves in agriculture. By making precise changes to plant DNA, we can enhance crop yields, making our food supply more resilient in the face of climate change. And then there's the creation of genetically modified organisms, or GMOs. With CRISPR, we can engineer organisms to produce useful substances like biofuels or pharmaceuticals that could revolutionize industries. The potential is vast, but it's not without controversy. As we wield the power to rewrite the code of life, we must tread carefully. Despite its potential, CRISPR also presents some significant challenges. Firstly, there's the risk of off-target effects. While CRISPR is precise, it's not perfect. Unintended changes to the DNA could occur, potentially causing more harm than good. Secondly, we've already touched upon the profound ethical concerns surrounding gene editing. The ability to alter life's code could lead us down a slippery slope, with consequences we can't fully predict or understand. Thirdly, there's the need for careful regulation and oversight. As we continue to unlock the secrets of our DNA and the potential of CRISPR, we must ensure that this powerful tool is used responsibly. These challenges are not insurmountable, but they must be addressed as we continue to explore the potential of CRISPR. The power to edit life's code also raises profound ethical questions. Imagine, if you will, the ability to design our offspring to eliminate genetic diseases or to enhance physical traits. This is the potential reality with CRISPR. However, the promise of human genetic engineering also presents us with a moral quandary. In our pursuit of progress, we may inadvertently open Pandora's box. The creation of designer babies could widen existing social inequalities, with genetic enhancements available only to those who can afford them. Furthermore, the long-term effects of gene editing are unknown. Altering one part of our complex genetic code could have unforeseen ripple effects, potentially creating new health issues or altering aspects of our humanity. As we stand on the brink of this brave new world, we must grapple with these ethical considerations. The power of genetic editing is in our hands, and with it comes great responsibility. As we harness this power, we must tread carefully. As we gaze into the future, the potential of gene editing seems limitless. CRISPR, as a revolutionary tool, holds the promise of transforming areas ranging from human health to agriculture. Imagine a world where we can not only cure genetic diseases, but also engineer crops to withstand harsh environmental conditions, thereby tackling global food shortages. However, the vast potential of this tool must be tempered with ethical considerations. As we stand at this crossroads, it's crucial to engage in public discourse about how we should and shouldn't use this technology. While the promise of gene editing is immense, we must tread carefully. The future is indeed exciting, but it's a future that must be navigated with wisdom and responsibility. As we continue to unlock the secrets of our genes, let us remember that the future of gene editing is in our hands. The notion of designer babies is perhaps the most contentious aspect of CRISPR. The concept of selecting and modifying genes in embryos to ensure certain traits in children is a polarizing topic. On one hand, CRISPR could potentially eliminate genetic diseases from a family line, offering hope to millions. Imagine a world free from the grip of conditions like cystic fibrosis or Huntington's disease. However, the flip side of this coin raises profound ethical questions. If we begin to edit genes for health, what's to stop us from editing for aesthetics or ability? Could we inadvertently create a society where children are valued for their engineered traits rather than their inherent worth? And what about the potential for unforeseen consequences down the line? These are not easy questions, and the answers are not clear-cut. As we continue to unlock the power of CRISPR, we must also grapple with these ethical dilemmas. The debate is far from over. Even with precise tools, there's always a risk of unintended consequences. CRISPR, as revolutionary as it is no exception. The potential risks of gene editing range from off-target effects to ecological impacts. Imagine the domino effect that one small alteration in a gene sequence could have. It could inadvertently affect non-target species, disrupt genetic diversity, or even alter entire ecosystems. This is the inherent risk of wielding such a powerful tool. It's a sobering reminder that a single change can ripple through an ecosystem. Despite the challenges, the future of CRISPR is promising. As we stand on the precipice of a new age in genetics, scientists around the globe continue to explore the vast potential of this revolutionary tool. Current research is pushing the boundaries of what we once thought possible, aiming to tackle some of the most persistent and challenging diseases of our time. From developing more effective treatments for cancer and HIV, to potentially curing genetic disorders like cystic fibrosis and Huntington's disease, the possibilities are truly staggering. Yet, with great power comes great responsibility. It is crucial that we establish and adhere to ethical guidelines as we navigate this new frontier. The potential for misuse is real, and the consequences could be far-reaching. we must ensure that CRISPR is used judiciously, with a deep respect for the intricacies of life that we are yet to fully understand. CRISPR is not just a tool, it's a responsibility. The power to shape life is now in our hands. With CRISPR, we're on the cusp of a new era in human history. This revolutionary technology gives us the potential to manipulate DNA, the building blocks of life, with an unprecedented level of precision. We can correct genetic diseases, enhance crop yields, and even create genetically modified organisms. But with this power comes an immense responsibility. As we wield this tool, we must also grapple with the societal implications. How will CRISPR affect our understanding of what it means to be human? What safeguards do we need to ensure that gene editing is used ethically and responsibly? These are questions that we, as a society, need to answer. We can't afford to be passive bystanders in this conversation. The decisions we make today will shape the future of our species and our planet. We're not just observers of evolution anymore, we're participants. The conversation about CRISPR is crucial, and it involves all of us. This isn't just about science, it's about our future. It's about the ethical choices we're making today that will shape the world of tomorrow. The power of CRISPR is immense, and so is the responsibility that comes with it. Take the time to educate yourself about this revolutionary tool. Engage in discussions, ponder its implications, and consider the ethical dilemmas it presents. Join the conversation and help shape the future of CRISPR. Let's quickly recap the key points we've discussed about CRISPR. We started our journey by understanding what CRISPR-Cas9 is, a revolutionary gene editing tool that allows for precise modifications in DNA. This powerful technology has a wide range of applications. It can potentially cure genetic diseases, enhance crop yields, and even create genetically modified organisms. We also took a deep dive into the ethical implications of CRISPR. As we navigate this new frontier, we grapple with profound questions. What are the consequences of human genetic engineering? How do we handle the controversy of designer babies? And then there are the unintended consequences to consider. While the potential benefits are immense, the risks cannot be ignored. We must tread carefully, understanding that each genetic alteration can have far-reaching effects. As we look to the future, we recognize that the power of CRISPR is immense, but so too is our responsibility. It's a brave new world we're stepping into, one where we hold the keys to our own genetic destiny. CRISPR is more than just a revolutionary tool, it's a paradigm shift. As we've journeyed through the intricacies of this technology, we've seen its transformative potential. It's a key that can unlock cures for genetic diseases, boost crop yields, and much more. Yet it also opens a Pandora's box of ethical dilemmas. As we harness the power of CRISPR, we must tread carefully, balancing innovation with responsibility. As we step into this new era, we carry the future of life in our hands. Thank you for joining us on this journey into the world of CRISPR. Your engagement truly matters, so don't hesitate to hit that like button, drop a comment, or subscribe for more enlightening content. Stay curious, stay informed, and help shape the future of our world.